Okay, I don't know what's going on with my hair, but hopefully it's entertaining to you all. Let's just get into today's lesson. This is legitimately like the fifth time I've tried to record this. Oh my god. There's been lots of technical difficulties, but we're gonna stay positive. We're just gonna keep moving. Our lesson objective for today is that students should be able to differentiate between different mechanisms of non-Mendelian genetics, including incomplete dominance, codominance, multiple alleles, and sex-linked traits. So let's review Mendelian genetics first. So unicorns can have differently shaped horns, either curly horns or straight. Assume that horn type is determined by a single gene and that the allele for straight horns is dominant over curly. In a cross between two curly-horned unicorns, what percentage of offspring do we expect to have straight horns? First things first, let's find our important information in the problem. So the problem already tells us that straight horns is going to be dominant, so a capital letter, while curly is going to be recessive, the lowercase letter. In the next sentence, the problem also tells us that we are crossing two curly-horned unicorns. That means that the mom is curly and the dad is curly. They're both curly organisms. The question is asking us how many of their offspring would have straight horns. So let's go ahead and set up our Punnett square. We're going to use the mom and the dad genotypes, and then we're going to find out the children's phenotypes. We all already know how to do this, but just in case you might have somehow not remembered how to do that, little h, 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 little h. They're all little h, which means they all have curly horns, which means no offspring have curly horns or have straight horns. So that was a thing that my in-person students, I realized this had happened. It's okay for the answer to be 0. A lot of times the answer could be 0. They may ask you a question knowing that the answer is 0, okay? So don't be afraid that if you're looking at your Punnett square and you feel like the answer to the problem is 0, that's perfectly fine. Just like in math class, sometimes answers are 0, okay? Recapping Mendelian genetics. So Mendelian genetics, there is this thing called dominant. Dominant is represented with a capital letter. If dominant is in the genotype, then it's going to be seen. All recessive alleles are going to be the weaker, smaller alleles that are only seen if there are 2 recessive alleles. This is what this might look like. So we might have homozygous, a homozygous, which are the same, heterozygous, where the things are different. So homozygous might be homozygous dominant, is represented by pink. The recessive allele could be represented by green, which means that a heterozygote would have 1 pink, 1 green, which could be represented by a dominant letter and a recessive letter. Okay, so this is what this looks like. So notice that homozygotes are the same to each other and heterozygotes are different from one another, different alleles. It's a recap, so I didn't go too much into it, but if you have any questions about that, let me know. So let's get into non-Mendelian genetics. So non-Mendelian genetics is exactly what it sounds like. It's the heredity that does not follow the normal rules that we've been using. So it doesn't follow normal dominant recessive rules, but you will still use a Punnett square. Just know that non-Mendelian genetics could include incomplete dominance, codominance, things that have multiple alleles, and even sex-linked traits. So incomplete dominance. Literally what it sounds like. It's dominant, but it's not completely dominant. It's not taking all the way over. It's going to take over a little bit. So incomplete dominance means that neither allele is dominant and neither allele is recessive. They're going to be somewhere in between. So an organism. So let's say that we know that inheritance of this petal color is incompletely dominant because they tell us in the problem, or we see it in the picture. One of the parents is CR, which just means red. The other parent is CW, which means white. Whenever we cross those and mate them, their offspring are going to be CR and CW. They're going to be pink. They're going to have a little bit of red and a little bit of white. So think of it like mixing paint. The red isn't completely dominant and the white isn't completely dominant. They're both equally dominant, which means they're going to mix together like paint. So let's do a practice problem. In a certain species of fish, blue scales are going to be completely dominant to yellow scales. So on my problem right here, I'm going to say, make it a little bit bigger. Blue scales are completely dominant to yellow scales. Cool. If you cross a blue fish with a green fish, how many offspring will be yellow? So let's go ahead and draw a Punnett square. It tells us that we are crossing a blue fish. Blue fish, as we know, big B and big B. Try and bring it maybe a little bit closer. With a green fish. So we have green fish, B and Y. How do you know that that's green? Well, I mean, in art class, when you mix blue and yellow, you get green. Perfect. Now we're going to fill in the offspring. We'll change colors. All right. So B, 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 Y, B, B, and B, Y. All right, so this is our parents. These are our offspring. So we know that B, there's 2 out of the 4 of those. Those are going to be blue. While B, Y, 1, 2 out of the 4, which are going to be, they're both blue and they're yellow, so they're going to look green. The question asked, how many offspring will be yellow? None. None of them will be yellow. So you'll say, none. None of them will be yellow. All right, and let me show you what that looks like. In case that wasn't very clear for you, that's fine. I put it also on here. So the first things first, you're going to put your parental genotypes that are given to you in the problem. Then you're going to figure out what goes inside. So B, 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 Y, B, and Y. Then you're going to determine what that genotype actually means. So these are blue fish, these are green fish, and then you're going to answer your question. None of the offspring will be yellow. Cool. That's incomplete dominance. Now let's talk about codominance. Codominance. Codominance is when both alleles are going to be completely dominant. They're not going to blend, such as in this example. They're not going to make, like blue is not going to blend with yellow to make green. Okay? Codominance means they're both going to be completely dominant, which means they're going to pop up full strength wherever they can. So they're both going to contribute to the phenotype equally. So in cows, there's things that are called roan cows, which means that in parts of them, the red allele is going to be activated. But in other parts of them, the white allele is going to be activated. So they're both going to be fully, completely dominant wherever they can be. Okay, it's called codominance. They're going to share dominance. So let's try an example together. All right, so while I'm taping this up, go ahead and read that problem to yourself. All right, let's get started. So, if black chickens are codominant to white chickens, show a cross between a black chicken and a spotted chicken, which is BW. How many offspring will be spotted? So let's write down our information. So we know that BB is a black chicken, We know the WW is a white chicken. And we know that BW chickens are spotted. They have white spots and black spots. They're also known as erminette. That's the fancy word for it. You don't need to know that. It's just a fun fact. So we're looking for how many offspring will be spotted. All right. So we're going to be doing a cross. So let's go ahead and draw our Punnett square. All right. And it tells us we're crossing a black chicken. So black is big B, big B. And a spotted chicken, B and W. Then we're going to find the offspring and see how many of them will be spotted. B and B. This box right here is going to come from here and from here. So B and B. Here, this box right here is going to come from this W and this B. So B and W. Here, it's going to come from B and W. Number one question that I get from students on these types of problems is, can I write the W first? You can write the W first. It's just I always choose one that I always write first just because it looks nice. But I mean, it's not wrong to say WB. All right. So how many of the offspring will end up being spotted? Well, this one and this one. Right? This is BW, this is BW. So 2 of the offspring out of how many total? 1, 2, 3, 4. 2 out of 4, which you should know is 50%, which is the same thing as 1 out of 2. These are all the same thing, which could also be represented as that, right? Half. Or if you were to drive it in 4s, 2 out of 4. Math. This is about as much math as I can do. So I like to show off a little bit. Oh, Jesus, that scared me. So this is what it looks like. Again, you're going to put the parent genotypes. Then you're going to fill in the offspring. Then you're going to say what the offspring's phenotypes would be, what would they look like. Then you decide which ones are answering the question. So it's pretty straightforward. Realize, though, that incomplete dominance and codominance are not the same. They're not the same. So let me show you how they're not. So in normal Mendelian genetics, complete dominance we've been doing for the past couple days, if there was a blue and a yellow, and blue was said to be dominant, then the offspring would end up being just blue because it would be completely dominant. Then we just learned about codominance and incomplete dominance. What is the difference? Well, if you have a blue parent and a yellow parent, in codominance, both blue and yellow are both going to be dominant. They're both going to be completely seen. Oh, go back. Oh my God. They're both going to be completely seen. You're gonna have blue spots and yellow spots. Versus in incomplete dominance, blue and yellow, neither of them are completely dominant. They're partially dominant. So if they're partially dominant, they're going to show a little bit, and yellow's going to show a little bit, and then they're both going to look, and it's going to end up looking green. Okay, so codominance versus incomplete dominance. So that's going to be the difference between those two. All right, multiple alleles. So in many cases, more than 2 alleles are possible for a single gene. So, so far we've been looking at, like, for example, pea shape. We were looking at yellow seeds or green seeds, right? For hair, we were looking at curly hair or straight hair, right? But oftentimes, life is not that simple. Life actually is going to be very complicated. So there's multiple alleles for a single gene. And the most famous example of that is blood typing, right? There are people who are type A. There are people who are type AB. There are people who are type A with an O carrier. There's type B with an O carrier, or there's people who are just flat out type O, right? So there's many, many, many different blood types. But remember, You as an individual only get two alleles. There might be 20 alleles out in the world, 20 alleles that are possible, but each individual only is going to get two of them. Okay. So the allele IA for blood typing is going to produce antigen A, which is going to give someone a blood type of A. The allele IB is going to produce antigen B, which is going to give someone a blood typing of B. The allele lowercase I is going to produce no antigen at all, which is what's known as O, type O, the universal donor. One of the most famous examples of this, besides blood typing, is also going to be like the fur of rabbits or the furs of different animals. They have all these different genotypes and they have all these different crazy rules. Luckily, at this level of biology, we keep it pretty simple. The most complex thing that we're going to do is probably blood typing, and even that you'll see is not that hard. So let's try it together. Let's try it together before you try it on your own. So let me type up another paper, BRB. All right, so we have a man with blood type AO and a woman with blood type B. What are the possible blood types of their children? So, let's go ahead and write out our blood types. We have IA, which gives you an A, IB, which gives you a B, and the little I, which gives you an O. Cool. Let's do our Punnett square. So we have a man with this blood type, AO. So we know if he's A, he has an I and an A. If he's O, he also has a little I. This one has a blood type B. She's just IB, IB. Cool. We're doing good. Now let's figure out in the middle. So right here, we're going to have IA, IB. Down here, we're going to have IB, I. Right here, we're going to get IA, IB. Still with me? Then we're going to have IB, I. I'm going too fast for you. Use that little, where is it? On this side. Use a little gear button down here and slow it down. Use a little gear button. Add whatever language you primarily speak. You can add it as a translation. Okay, so you can try and help you. All right, boom, cool. What are the possible blood types of their children? Well, let's see. We have a child that is IA, IB. So that is going to be AB. One child. Oh, there's two of those. It's two of them out of the 4. 50%. Okay, let's see what's our next genotype. I have IBI, which is going to give us B, or you could call it BO. There's 2 of those out of the 4. That's 50%. So what are the possible blood types of their children? A, B, and B. Let's show what that looks like. So again, we put our parental genotypes. We fill in our squares. We determine what those phenotypes would be, and then we answer the question. So that's multiple alleles. All right, we're almost finished. Sex-linked traits. So what is a sex-linked trait? Well, a sex-linked trait occurs when a trait is found on the X chromosome. These conditions occur more often in males than in females. Why? Well, let's look right here. This is women and men. Women have 2 X chromosomes. Men have an X chromosome and a Y chromosome. So why would we think that a defect or some sort of trait that is found on the X chromosome would affect males more than it would females? Well, because if females get a bad X on one chromosome. The other X could be dominant and hide the bad one. Versus if a male gets a bad trait on their X chromosome, there's no other X chromosome that could possibly win. This is the only one that they've got. So if it's bad, then it's just bad. Okay, this is another way of looking at the same thing, just a little bit bigger. So let's say that there's a recessive gene that is located on the X chromosome. It gets blocked by one that is a dominant one. Over here, this is XX, this is a woman. So by chance, she did get a recessive allele, but luckily, she had another X chromosome that had a dominant allele that actually masked that recessive. So the woman is unaffected. However, right here, let's look at the same situation, but on a male. So they have an X chromosome, they have a Y chromosome. The X chromosome, let's say, has a recessive gene that causes some sort of disease here. Unfortunately for the man, he doesn't have another chromosome that can dominate this one, which means that X trait, they've just got to have it. It's just displayed, even though there's only that one. And that's why males are more affected by sex-linked traits than women. So again, a sex-linked trait is going to be a trait that is found on the X chromosome. The normal is typically going to be dominant, and the affected or the diseased or whatever you want to say is going to be recessive with an H. While a carrier is going to be a person with a normal trait that displays the normal trait, but could potentially pass it on to their offspring. That was that example right there that we had on the last page. Go back to it. This person right here would be called a carrier because they display normal, right? They display the dominant, which is the normal, but they do have the potential to possibly pass on that recessive gene to one of their children who could then be affected. So this is what this kind of will look like. Okay, notice that mom is going to have XX, dad is going to have XY. Notice there are no alleles on the Y because we're looking at a sex-linked trait that is found on the X chromosome. Also notice that H, the big letter, the dominant, is unaffected, while the small letter is showing affected. Let's do a problem together. All right. A colorblind woman and a man with normal vision want to know the chances that their son will have normal vision. First things first, we always want to determine what our different things are. So color blindness is going to be represented by the recessive trait. So that is color blind. While X big B is going to be not color blind. Okay, and then we're going to do our Punnett square. So a colorblind woman, so she's X. So what I always like to do mom, dad, then assign their whatever it says. Colorblind woman, so mom is colorblind, which means she must have both recessive. And a man with normal vision, which means that he has the dominant trait. Want to know the chances that a son will have normal vision. So let's look. We have X and X. So in this box right here, try and really show this good. So this is usually where kids will get confused. So right here in this box, X and X. X big B, X little b. Next one, X little b, Y. Still with me? Right here, X big B, X little b. Last box, X little b, Y. Okay. Now I want to point out something. Seems obvious, but you might accidentally forget. XX, these are girls. These are boys. Now we're going to say if they're colorblind or not. For this girl, she has a big B and a little b. Will she be colorblind? No. X big B means that they're not colorblind. So she's not colorblind. How about this girl down here? X big B and a little b. She has a big B, which means that she is not colorblind. How about this boy? Let's look at him. X little b. Will he be colorblind? Yes, he will be colorblind. X little b. This boy will also be colorblind. Now the question is asking, what are the chances that a son will have normal vision? So, we're looking for chances. We're going to say blank sons out of blank are going to be normal. How many sons total are there? There's one boy and there's two boys. So we have two boys total. How many of these boys are colorblind? 1 and 2. So 2 out of 2 sons. Oh, it's asking are normal. Sorry. How many of these are normal? None. 0. So 0 sons out of the 2 sons are normal. Do not mess this up. If the problem asks about sons, we're going to ignore the girls. We're not looking at the girls. The problem's not asking me about girls. I'm not going to talk about them at all. The problem asks about the sons. So I'm asking about the sons. I'm only looking at the boys. So how many of these 2 are going to be normal? None. Now, if the problem were to somehow say, what are the chances that their child will have normal vision? Then that's where we look at boys and girls. Okay? So that's where the reading is key in these types of problems. I mean, reading is literally 100% of it because the Punnett square is so easy. Anyone can do that. The difficult part in these problems is going to be reading the problem and seeing what are they actually asking me? What are they actually asking me? And if you're curious, this is what color blindness. If you cannot distinguish these letters or these numbers from their surroundings, then you might be colorblind, and that's okay. Another thing that is characterized by sex-linked things that people don't think about is male pattern baldness. You would assume that if your dad is bald, that you would be bald, but male pattern baldness is actually inherited on the X chromosome, which means you're going to get it from your mom because you get your Y chromosome from your dad, you get your X chromosome from your mom. So if your mom's male relatives are going bald, then you will go bald as well. Fun fact. Okay, so again, for this problem, we're going to put our parental genotypes. So we have mom over here, we have dad over here. We're going to fill in. We're going to recognize that we have females and males, and then we're going to see they're colorblind or not colorblind. Then we determine how many of the sons will actually have normal vision. So 0 out of the 2 sons will have normal vision. Cool. All right, so that wraps up the end of this. So you're going to finish up your practice problems, do your exit ticket, and as always, stay awesome, stay alive, stay cool. I'll see you guys later.